Hello dear students, now we'll learn about hematopoiesis. So the definition of hematopoiesis is okay, the cells originate in the bone marrow from a common stem cell. The process of generation of these cells is termed as hematopoiesis and the stem cell is called as hematopoietic stem cell. So what is hematopoiesis? It is the process of generation of the blood cells. So this process occurs, where does it occur? So this process occurs in bone marrow. So it is the location where the hematopoiesis occurs. So in order to understand what is hematopoiesis, first we learn about some terms called as progenitor cells, the stem cells, the other kind of the cells that we have to learn in order to understand the process of hematopoiesis. So first is, what are progenitor cells? Every cell in the human body and that of other mammals originates from stem cell precursors. So we need to remember that in the human body are other mammals like other vertebrates also. So where does the stem cells originate? Okay, they originate from the stem cell precursors. So the progenitor cells are what then? They are descendants of stem cells that which further differentiate in to create the specialized cell types. So there are two kinds of the cells here. The first kind we are learning is the progenitor cells. So the progenitor cells originate from the stem cells. There are many types of the progenitor cells throughout the human body. Each progenitor cell is only capable of differentiating into cells that belong to the same tissue or organ. ಅಂದರೆ ಯಾವುದೇ ಒಂದು progenitor cell, it can give rise to the cells that belong to the same type of tissue ಅಥವಾ ಆರ್ಗನ್ ಅಂಗ. ಸೊ ಈ ರೀತಿ ಒಂದೇ ಟೈಪ್ ಆಫ್ ಸೆಲ್ಸ್ಗೆ ಸೆಲ್ಸನ್ನು ಕೊಡುತ್ತೆ, ಅದೆಲ್ಲ ಪ್ರೊಜಿನೇಟರ್ ಸೆಲ್ಸ್ ಆಗುತ್ತೆ. Some progenitor cells have one final target cell that they differentiate to, while others have the potential to terminate in more than one cell type. So now let us see what exactly is the difference between the progenitor cells and the stem cells. So we know about the progenitor cells now. Now the stem cells share two qualifying characters. ತುಂಬ ವಿಶೇಷ ಗುಣಗಳಿದೆ ಸ್ಟೆಮ್ ಸೆಲ್ಸ್ಗೆ. ಏನದು? ಫಸ್ಟ್ಲಿ, ಆಲ್ ಸ್ಟೆಮ್ ಸೆಲ್ಸ್ ಹ್ಯಾವ್ ಪೊಟೆನ್ಷಿಯಲ್ ಟು ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಇಂಟು multiple types of cells. So you might have heard about the stem cell therapy. So these are the very basic kind of the cells which can give rise to other kinds of the various other kinds of the cells. Secondly, what is the second character? Stem cells are capable of unlimited self-replication via asymmetric cell division, a process known as self-renewal. That means they have the capacity to give rise to more number of the same kind of the stem cells. That means they can go for infinite number of the synthesis of their own cells, and this is called as self renewal. So the two important characters of the stem cells are differentiation and self renewal. There are two broad categories of stem cells found in all mammals. Okay, so the two kind of the stem cells are, the first are the embryonic stem cells. What are embryonic stem cells? Okay, these arise these cells arise from the inner cell mass of the blastocyst in an early stage of the embryo. Embryonic stem cells are blueprint used to create every cell in the body. Remember students, the embryonic stem cells are very, the master copies like, so which can give rise to the every cell of the body. because they can be used to create any kind of the cells. They are known as pluripotent. That means they have the capacity to uh, give rise to the infinite number of the cell types. That is why they are called as pluripotent. Then the second type of the stem cell are the adult stem cells, are the somatic stem cells. Okay. uh, Unlike the pluripotent embryonic stem cells, adult stem cells are more limited in relation to the type of the cells that they become. So, the adult stem cells, they give rise to the limited number of the other kind of the cells. Unlike embryonic stem cells that could be used to create any cell, so embryonic stem cell ಇಂದ ಯಾವುದೇ ಟೈಪ್ ಆಫ್ ಸ್ಟೆಮ್ ಸೆಲ್, ಯಾವುದೇ ಟೈಪ್ ಆಫ್ ಸೆಲ್ಲಿಂದ ನಾವು ಯಾವುದೇ ಟೈಪ್ ಆಫ್ ಸೆಲ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ. ಬಟ್ ಆದರೆ ಅಡಲ್ಟ್ ಸ್ಟೆಮ್ ಸೆಲ್ಸ್ ಆರ್ ಲಿಮಿಟೆಡ್ ಇನ್ ಜನರೇಟಿಂಗ್ ದ ಸೆಲ್ ಟೈಪ್ಸ್ ವಿತಿನ್ ಅ ಸ್ಪೆಸಿಫಿಕ್ ಲೈನೇಜ್, such as blood cells or the cells of the central nervous system. This level of differentiation is termed as multipotent. Remember students, whereas the embryonic stem cells are pluripotent, the adult stem cells are multipotent. When compared to stem cells, the progenitor cells have lesser ability to self-renew. ತಮ್ಮನ್ನು ತಾವು ಮತ್ತೆ ಉತ್ಪತ್ತಿ ಮಾಡಿಕೊಳ್ಳುವಂತಹ ಸಾಮರ್ಥ್ಯ ಸ್ವಲ್ಪ ಕಡಿಮೆ ಆಗುತ್ತೆ. A stem cell self replication is indefinite. ಎಷ್ಟು ಬಾರಿ ಬೇಕಾದರೂ ಅದು ಸೆಲ್ಫ್ ರೆಪ್ಲಿಕೇಶನ್ ಆಗ್ಬೋದು ಸ್ಟೆಮ್ ಸೆಲ್, while that of the progenitor cells is not. Additionally, the cell potency of the progenitor is usually more restricted. While some progenitor cells are multipotent, none are pluripotent. ಯಾವುದು ಕೂಡ ಪ್ಲೂರಿಪೊಟೆಂಟ್ ಅಲ್ಲ, ಯಾವುದೇ ಪ್ರೊಜೆನಿಟರ್ ಸೆಲ್ಸ್ ಕೂಡ ಪ್ಲೂರಿಪೊಟೆಂಟ್ ಅಲ್ಲ, ಮಲ್ಟಿಪೊಟೆಂಟ್ ಆಗಿರಬಹುದೇ ಹೊರತು ಪ್ಲೂರಿಪೊಟೆಂಟ್ ಯಾವುದು ಕೂಡ ಇರೋದಿಲ್ಲ, ನೆನ್ಪಿಟ್ಕೋಬೇಕು. Other progenitor cells are only capable of differentiating into one cell type known as unipotency. Now we'll understand about the hematopoiesis. So this is the hematopoietic chart. So this process occurs in bone marrow. Let us see the process bit faster. Hematopoiesis is the process by which all blood cells are produced and occur in the bone marrow. Bone marrow stromal cells produce cytokines which guide the process of the hematopoiesis based on the demand and supply. In the bone marrow niche, there are stem cells which have a potential for self-renewal as well as differentiating into progenitor cells. Stem cells of bone marrow give rise to two types of the progenitor cells. We can see there are two kinds of the progenitor cells here. The hematopoietic progenitor cells, what are they? So the hematopoietic progenitor cells are an intermediate cell type in blood cell development. Hematopoietic cells are immature cells that develop from hematopoietic stem cells, as that these cells can both self-renew and differentiate into hematopoietic progenitor cells. The hematopoietic cells eventually differentiate into one of more than 10 different types of the mature cells. You can see here students, so there are various kinds of the cells finally that are produced through the process of hematopoiesis, like thrombocytes, that is platelets, or the erythrocytes, mast cells, the basophils, neutrophils. Like this, there are various kinds of the cells that are produced finally through the process of hematopoiesis. These are majorly the immune cells. You can see the hematopoietic stem cell, which is also called as hemocytoblast. It gives rise to the two kinds of the progenitor cells is, the first one is the myeloid progenitor, so which is called as common myeloid progenitor, and the second kind is the lymphoid progenitor. So this is the common lymphoid progenitor. So this is one lineage and this is another lineage. So let us look into the common myeloid progenitor. So while we learn about the common myeloid progenitor, if you look at this one, the first kind of the cells that are given by this common myeloid progenitor are the megakaryocytes. So the the platelet precursors are called as megakaryocytes, which terminally differentiate to release the platelets. Megakaryocytes are rare cells confined to bone marrow and therefore difficult to harvest in sufficient numbers for laboratory use. The number is very low. Thrombopoiesis is the formation of the platelets in the bone marrow. You can see here, students, that the megakaryocytes give rise to thrombocytes. So the thrombocytes are nothing but the platelets. So through the process called as thrombopoiesis, the platelets are produced. So this is about the platelets. The second kind of the cells that are produced from the common myeloid progenitor are the erythrocytes. So the erythrocytes life cycle involves three stages: production, maturity and destruction. Okay, thus production, maturity and destruction. So these are the three stages. Through the erythropoiesis, which is the production of erythrocytes, a sub process of hematopoiesis occurs in the bone marrow. So the process of synthesis of erythrocytes or the production of erythrocytes is called as erythropoiesis. The lifespan of these erythrocytes is approximately how much? It is 120 days, after which they die. The primary role of these cells is to carry oxygen from lungs to other parts of the body, while they also carry back the carbon dioxide from other parts of the body to the lungs for the exhalation. So the next kind of cells are the mast cells. You can see, students, mast cells. Mast cells are located in areas uh, below the epithelium in connective tissue surrounding the blood cells, smooth muscle, mucus and hair follicles. These are among the first cells to interact with the environmental antigens and invading pathogens or microorganisms. These are produced from the bone marrow and are released into the blood as undifferentiated cells. Remember, they are not matured in the bone marrow, but they are released as undifferentiated cells, and these cells then mature after they leave the blood and reach the specialized tissues. Mast cells are primarily involved in IgE dependent responses. So they are involved in collaboration with the IgE dependent responses. The next kind of the cells are the myeloblast. So it is this stem cell is differentiated into the effectors of the granulocyte series. Stimulation of myeloblast by GCSF and other cytokines triggers the maturation, differentiation, proliferation, and cell survival. So the first kind of the granulocytes that are produced from the myeloblast are the basophils. What are basophils? You can see basophils are type of the white blood cells. Like most types of the white blood cells, basophils are responsible for fighting fungal and bacterial infections, and even the viruses. They are granulocytic cells, which means they release granules of enzymes to fight against the harmful bacteria and the germs. Basophils, neutrophils, and you can see that is basophils, neutrophils, and the eosinophils are the three types of the granulocytes in the body, each of which release different kinds of the enzymes to fulfill the different role in the body's immune response. The enzymes that basophils release are called histamine and heparin. So, whereas histamine is a vasodilator which widens the blood vessels near an infection to open and allows more blood to flow to the site of the infection. Whereas heparin is a naturally occurring blood thinning substance which prevents the clotting. Basophils are responsible for the body's immune response during allergic reactions. When the body is exposed to allergen, basophils release histamine, which triggers the physical symptoms of allergic reaction. So this causes the typical allergic reactions like the running nose and the watering eyes, etc. The second kind of the granulocytes that are produced from the myeloblast are the neutrophils. So these are the dedicated mac- these are the dedicated phagocytes. Neutrophils are dedicated phagocytes which perform only the function of the phagocytosis, that is engulfing. They are smaller than macrophages. You can see these are neutrophils, whereas these are macrophages. So the size of the neutrophils is smaller than the macrophages, hence these were earlier called as microphages. So they have plenty of lytic granules. They have plenty of lytic granules in their cytoplasm and have irregularly shaped nucleus, because of which they are also known as polymorphonuclear leukocytes. In circulation, they have very short half-life. Remember students, the half-life of the neutrophils is very less, so it is less than 2 days, because of which a large reservoir of neutrophils is kept in the bone marrow to be released as soon as the infection is sensed. The next kind of the cells are eosinophils. They have bilobed nuclei. You can see they have bilobed nuclei. Cytoplasmic granules of eosinophils take up the acidic stain of eosine red, and hence they are called as eosinophils. Similar to neutrophils, these cells also migrate to the site of the infected tissue. However, they have weak phagocytic activity. Eosinophils are supposed to play a significant role in the parasitic infection. So the monocytes, they are the type of the leukocytes, are the white blood cells. They are the largest type of the leukocytes and can differentiate into macrophages and the dendritic cells. Monocytes and their macrophages and dendritic cell progeny serve three main functions. Which are the three main functions? So they are phagocytosis, antigen, that is antigen presentation, and cytokine production. So now I am coming to the second lineage of the progenitor cells, that is common lymphoid progenitor. So this is the lineage that is from the common lymphoid progenitor. Now let us look into the first kind of the cells that are produced from lymphoid lineage are the natural killer cells. The natural killer cells are a group of innate lymphocytes that have catalytic activity against virus infected cells and physiological stressor cells like tumor cells. The name natural killer cells is derived from the fact that these cells can target cells without the need of the prior activation. Remember students, they do not need activation, and they naturally you know they attack the target cells. These cells are responsible, essential for the management of immunological responses as well as for the innate immunity. Natural killer cells secrete cytokines like interferon gamma, TNF alpha, and the chemokines. Catalytic activity of natural killer cells is one of the most important responses of natural killer cells against antigens. The activity is mediated by granules found in the cytoplasm of lymphocytes. Natural killer cells are also involved in the antibody dependent cell mediated cytotoxicity, whereas antibodies bound to antigens are recognized by the specific receptors that are present in the NK cells. The one more kind of the cells are that are produced, that is relatively the natural killer cells. They are the large granular lymphocytes, whereas the small lymphocytes, they give rise to two kinds of the lymphocytes. One is T lymphocytes and the other is a B lymphocytes. Let us look into T lymphocytes. T cells also get their name from the site of maturation, that is thymus. So this is the location, that is thymus is the location where the T cells get matured, and hence they are given the name T, T for that. T cells also have surface receptors to recognize antigens, but they do not directly bind to the antigens like surface antibody receptors on B cells. That means they cannot directly bind to the antigens as B cells, but they have surface receptors to recognize antigens, but remember students, they do not directly interact with the antigens like B cells. These receptors recognize antigens bound to a special kind of the cell membrane protein called as major histocompatibility complex or MHC molecules. Lymphocytes differentiate into two main types, which are the helper T cells, cytotoxic T cells, and in addition, even memory T cells are also produced. So the memory T cells are antigen specific T cells which have longer lifespan. They play a key role in rapid immune response and the re-exposure to infectious agent. They are responsible for the secondary response. That means they have the capacity to remember which was that which pathogen had attacked previously, so that the uh, subsequent attack by the same pathogen is remembered by the memory T cells, and it is ready with the heightened immune response and quick response also. The B lymphocytes. The B cells get their name from the site of maturation in the birds where they were first discovered, that is Bursa fabricius. So that they are called as B cells. Whereas in humans and other mammals, the main site of B cell maturation is the bone marrow. The mature B cell synthesizes and expresses the specific antibodies produced in response to antigens. It binds to the specific antigen by the membrane bound immunoglobulin or the antibody. The activated B cells further they differentiate into, so you can see, further they differentiate into plasma cells or the effector cells. They lose the surface antibody and start producing the specific antibodies in a large number to fight the infection. B lymphocytes produce antibodies and hence they are known to trigger the humoral immune response. Okay. So like we have memory T cells, we also have that is B memory cells that are formed after the primary infection, and they remain in the blood for decades. They circulate in the blood, identify and act against previously infected antigens. So this is the overall view of the hematopoiesis. Hope you understood, friends. Thank you. Have a good day.